Maybe you can hit, okay, the recording button, yes. So can you see my screen?
Yes.
Good. Thank you. So I will be talking today about our work on applying deep learning to understanding of enzyme function. And I think in this community, I don't need to emphasize that plants are remarkable chemists, because obviously they can only rely on chemistry for their defense and adaptation to the environment. And as a result of 400 million years of evolution, they have learned how to produce a huge variety of specialized metabolites that sometimes turn out to be useful for us humans as medicines, for example, due to their bioactivity. So from this review, I would like to highlight just two compounds: artemisinin, the first-line antimalarial drug that was rewarded, or the discovery of which was rewarded with a Nobel Prize to UU2. And I want to point out that the artemisinin therapy market is worth hundreds of millions of dollars per year. And then another example, paclitaxel, which is the most successful chemotherapy drug derived from plants. And the paclitaxel market is worth billions of dollars per year. So these are also very valuable compounds. And as you can see from the chemical structure of paclitaxel, the chemical structures can be quite complex and not easily accessible by organic synthesis. So in recent years, there is a trend that we try to use the approaches of synthetic biology to gain access to these compounds from plants. So essentially starting from a medicinal plant that produces some unique valuable molecules, we use modern scientific approaches like transcriptomics, metabolomics to elucidate the biosynthetic pathways of these molecules. And once we know the biosynthetic pathway, we can engineer these enzymes into a laboratory microorganism such as yeast. And then we can use this yeast to produce the original molecules or potential derivatives of those molecules, ultimately arriving to molecules that are inspired by nature and can be used to develop new drugs without doing any organic synthesis at all. However, this sounds simple, but in practical applications, this part of elucidating the biosynthetic pathway is actually quite difficult in plants. So plants are extremely diverse. There's more than 400,000 known plant species. There's more than 200,000 known metabolites, but this is just a tip of the iceberg that we have annotated so far. And more importantly, the biosynthetic pathways are not encoded into gene clusters, or generally not encoded into gene clusters like what we see in microbes. And therefore, finding out the right biosynthetic enzymes that are connected into a pathway is quite difficult. And although the amount of sequencing data that we are obtaining on plants is increasing exponentially, our understanding of these enzyme sequences is not increasing exponentially. So in my view, we really need better computational approaches to understand what these sequences mean. And so the central question in my lab is how can we take advantage of modern computer science approaches to tackle this complex biochemistry of plants? And more specifically for this talk, the question would be, can we understand the function of a single biosynthetic enzyme directly from its amino acid sequence using deep learning? And for this project, we decided to focus on terpenoids. So terpenoids, as you probably know, are the largest class of natural products. And the reason why we selected this class is because in a way, their biosynthesis is pretty well understood, and it always starts from the same precursor molecule. So the biosynthesis of terpenoids starts from linear molecules that are composed of isoprene units and a diphosphate in the end. depending on the length of the precursor. We talk about monoterpenes, sesquiterpenes, diterpenes, and so on. There are longer terpenes as well. And in the initial step, these precursor molecules are cyclized into scaffolds that we can call terpene scaffolds by terpene synthases. And then these scaffolds are further decorated by additional tailoring enzymes such as cytochrome P450s, metal transferases, and so on, to produce the huge variety of terpenoids that we know already. And currently, the reviews mentioned there's over 8,000 known structures of terpenoids. These also include artemisinin and paclitaxel, which I mentioned in the beginning. So in this project, we will focus on the first enzymes that catalyze the first step, the cyclization of the isoprenoid diphosphate precursor, so terpene synthases. I should also mention that the reactions catalyzed by terpene synthases are quite complex. So initially, the linear substrate is formed into a certain shape in the active site of the enzyme, and then the diphosphate group is released and forming a cation. And then there is a chain of events that happens in the active site, which leads to multiple bonds being created or eliminated, and multiple rings being formed. And essentially, the molecule that then leaves the active site looks quite different from the original precursor. And I think this is what makes this problem also quite interesting from machine learning perspective. It is not a simple reaction such as addition of one methyl group. Okay, so what we would like to achieve. If we start from a protein sequence of unknown function, we would like to have a deep learning model that I depict here with this R2D2 that would tell us this sequence encodes a sesquiterpene synthase, which takes exactly this substrate and produces exactly this product. But this is something that no bioinformatic tool can do at the moment. And why is it not easy? Well, because we are dealing with huge spaces on both sides. So the sequence space, if we think of an amino acid of 500 amino acids long enzyme, it's a 20 to 500 possibilities, which is an infinitely huge number, much more than the number of atoms in the universe. So it's a huge sequence space, and here we have a huge chemical space. Also, the chemical space, even if we talk about molecules that are less than 500 Dalton, it's also an infinitely huge space. The biosynthetic reactions can be quite diverse, and the amount of data that we have is not quite at the scale that's typically used for deep learning. So nevertheless, this doesn't stop us, and we still try to make some progress here. And I want to show you why I personally believe that deep learning is really, really powerful. Maybe you have heard of things like AlphaFold, which are very relevant for biology, but I want to show you something different. So in case you haven't seen, this is the DAL-E model that was developed by OpenAI, and it's a model that can generate images based on text descriptions. But by text descriptions, I don't mean something simple, like give me a picture of a house, but it can be very elaborate text descriptions, such as here. Teddy bears mixing sparkling chemicals as mad scientists in the style of a 1990s Saturday morning cartoon. And if you give this description to the model, it generates an image that fits this description. And because this is a generative computational model, you can just ask it to give you another image and another image and another image and so on. So in my view, the level of creativity and deep understanding of the context of the text, the meaning of different words, is really, really powerful here. And if we want to apply something like this, obviously this was trained on hundreds of millions of images and text descriptions. So first we need to know how many terpene synthases do we actually have. And obviously there is no database out there of terpene synthases, so we had to start by building our own database. And this is mainly work of my bioinformatics student, Teresa here. We started building our own database, and this is something that's still ongoing. We still keep finding more enzymes in the literature. But after about 2 years of work, we now know that we have about 1,200 characterized terpene synthase enzymes, which catalyze about 2,400 different reactions. Most of these belong to the class of sesquiterpene synthases, and then we have monoterpene, triterpene, diterpene syntheses, and so on. If we look at the species distribution, the vast majority of them are found in plants, which is well known that the terpenoid biosynthesis has to the most extent radiated in plants. And then we have fungi, bacteria, and other organisms, including recent terpene synthesis discovered in marine organisms such as corals. If we look at the characteristics of these enzymes, we see they are quite diverse, ranging from 300 amino acids all the way to 1,000 amino acids. And depending on the kingdom, the distributions are quite different. So, for example, monoterpene synthesis in insects can be 350 amino acids, while in plants, they're typically over 600 amino acids. And also we can look at the sequences from the bioinformatics point of view and look at which protein domains, PFAM domains, can we find in these sequences. And the most common combination here is a combination of the terpene synthase C-terminal domain and terpene synthase N-terminal domain. But there are many other possible structures of these enzymes. And surprisingly, we found that some of the characterized TPS domains actually have TPS enzymes have no PFAM domains at all. So they're essentially invisible from the bioinformatics perspective. So these are the characterized terpene syntheses. How many uncharacterized terpene syntheses are out there? So for this, we did just a quick scan of the different sequencing databases, including UniProt, Phytosome with the plant genomes, 1KP with the plant transcriptomes, and other transcriptomes. Altogether, about 600 million protein sequences that we found. And we scanned them using the PFAM domains, which we know are not perfect, as I mentioned at the last slide. But for majority of the terpene synthesis, we found at least some PFAM domains characteristic for them. So this is good enough. And with this approach, we found about 200,000 putative terpene synthase sequences of unknown function. So this is a good number that it would be useful to characterize them. And I mentioned before that to apply deep learning seriously, we need hundreds of millions of data points, and we clearly are not at that number. However, there are approaches in machine learning that can deal with this, and the approach that we're using is called self-supervised learning. What that means? It means you can start with a large data set of uncharacterized data, such as UniProt, where we have 250 million protein sequences. And you start training a model that is supposed to solve a proxy task. So in this case, what we do is we hide certain amino acids, and we ask the model to predict what are the missing amino acids. With this approach, you can keep training for a long time on a large number of sequences, and the model has to essentially understand what it means to be a protein. It has to understand how these amino acids are related to each other. In a way, it also has to understand what would be the structure of the protein so that it understands how the amino acids are related. And so when you do this for long enough, you arrive to a model that sort of understands the proteins. And this cannot still do anything useful, but we can then take this model and then we can train it on a smaller data set, such as our terpene synthesis, in a supervised way, and then we arrive to a model that can perform some specific tasks. The nice thing is that all this training is actually something that we don't need to do ourselves because people have already done this. And there are several of these large pre-trained protein models available. We have tested all of them, and so far we have been most successful using this ESM1B model that was developed by Facebook. And we start with this model and we further fine-tune it using our data set of 200,000 uncharacterized terpene synthase sequences. So this improves the performance of the model slightly. It's not a dramatic improvement, but it improves slightly. And I would like to mention here that these large language models that are also used for the protein sequences are based on the transformer architecture, which is an architecture for deep neural networks that was introduced in 2017 and made a huge, or led to a huge progress in many areas, including language understanding and image recognition, and also biological applications. And this is based on many components that have been known before, such as feed-forward networks, residual connections in the modules, and encoder-decoder architecture, which means the model can generate an internal representation of the data and then decode the internal representation back to the original space. But the main innovation here in the transformer is called attention, and it has these attention modules, which essentially track the context between the different parts of the data. So to give an example, in language understanding, you can have a sentence like the animal didn't cross the street because it was too tired. And the model keeps different layers of attention between these different tokens. So it can understand that, for example, this word, it, is mostly related to the animal and not so much related to the street. And then it can keep this attention at different layers of abstraction. So this is obviously very important for protein sequences where each amino acid is related to other amino acids to a different degree. So now we have this transformer model for proteins. So let's go to an actual prediction task. And the first task we want to solve is detect terpene synthases and predict what is their substrate. So given an enzyme sequence, can we say whether it is a terpene synthase? And if yes, what is the substrate? Is it a GPP, FPP, GGPP, and so on? And this is mostly work of my PhD student, Raman, here. The good news is that there is already a tool exactly for this purpose, and that's called Terzyme. It was published a couple of years ago. And this tool is using hidden Markov models. So what this means, hidden Markov models are traditional bioinformatic tool. If you have a database of sequences, you can train a profile hidden Markov model, which looks something like this. This is showing DNA sequences here, but it works exactly the same for protein sequences. It sort of builds a model of probabilities based on where you are in the sequence, what might be the next residue, and how the sequence might branch, and so on. And if you have this kind of model, then for a new sequence, you can make a prediction how well it corresponds to this model. So is it part of the class or not? So using this Terzyme model, we have at least something we can compare against. But we have spent quite a long time thinking how to best evaluate these different approaches. So the classic approach in deep learning is using k-fold cross-validation. So that means you take all your data and you split it into some portion. In our case, we use 20%, which you hide from the model. This is called the test data. And the remaining data, the 80%, you use for training your model. And then after the model is trained, you evaluate its performance on the test data. And then you do this in one iteration, and then in next iteration, you select different data for the test fold. You train your model on the other part, and so on. You repeat this, and then you calculate some average performance based on all the iterations. But if you think about terpene synthesis, what could happen is that you might have, for example, a taxadiene synthase in here, and then you might have another taxadiene synthase from different species, which ends up here. And then the enzymes might be quite similar. And in that case, the task is going to be quite easy for the deep learning model. So that your performance evaluation is not going to be realistic. So I think the random splitting of the data here is not appropriate to get a realistic evaluation. So instead, what we try to do is we make a network of all the enzyme sequences that we have, where where the two enzymes are connected if they share at least one product. The terpene synthesis can have multiple products, so we get quite large networks. And on this network, we perform an algorithm that's called community detection, which highlights these clusters of enzymes that have the most similar product spectrum. And we make sure that when we split the data into the test folds and the training folds, we always make sure that each cluster is in a single fold. So that way we don't make the task too easy for the model. Okay, so we train the model, including the hidden Markov based approach similar to Terzyme on the same data, and we evaluate using this approach. And this is how the results look like. So here we have the performance of the Terzyme-like hidden Markov model approach. So this is our baseline. And then next to it is something I would call the naive approach using BLAST. So you just take your input sequence and you blast it against your data set of terpene synthases. And if the first hit is a sesquiterpene synthase, then you say your sequence is a sesquiterpene synthase. And it turns out that this naive approach actually performs slightly better than the baseline approach, which is funny. And then here we have different architectures of the transformer networks combined with different classifiers to classify the classes of the terpene synthesis. So far, we have found that the transformer combined with a random forest classifier performs the best. And as you can see, it's a huge performance boost compared to the baseline approach. So that is nice. And the question is, can this be useful for anything? So we try to test this also in the lab. And we start with 250 million sequences from UniProt. We scan all these sequences using our terpene synthase predictor, but we only select those which have not been recognized by PFAM. So they don't have any of the PFAM domains which are found in terpene synthesis. So we're looking for those terpene syntheses that have so far been invisible to the classic bioinformatics classification. And then we take those sequences, we do codon optimization for yeast, we synthesize the genes, put them into plasmid and express them in yeast, and we measure by GCMS if they actually produce any terpenes. So this is mostly work of my postdoc Theo here, and he's using this yeast cloning toolkit that was published by the Druber Lab, which is a super useful tool for synthetic biology, where you design components and you can combine these components in a single Golden Gate reaction into plasmids. And so you have all these components, including a common promoter and terminator and a yeast marker. And we just put in the terpene synthase enzyme that we want to test and make a plasmid very quickly. And we test these plasmids in two different strains. So we have one strain that was developed by Jay Kiesling's lab for diterpene production. It overproduces GGPP. And another strain from Joe Chappell's lab that overproduces FPP and is used for sesquiterpene production. And this is just one example. We so far tested about 20 sequences, and we found that about half of them actually do produce terpene products, which is very nice. And this is a very strange sequence from some Ercon that came up, and that actually produces several potentially novel sesquiterpenes here. We are now trying to figure out what these molecules are. And here I'm showing a clustering of GCMS spectra of sesquiterpenes by their cosine similarity. And here we place those 5 different products of this enzyme in the context of the known library spectra. And so we can potentially take a look at what it might be most similar to. Nevertheless, the similarity to the known spectra is not so great. So here we have the best match in the NIST database with cosine similarity about 0.7, and the best match in the Wiley database with cosine similarity about 0.45. So as you can see, the spectra don't really look too similar. So I think there is a good chance that we have actually discovered completely new sesquiterpenes here. And now we can move on to the second task, which is the prediction of the actual product of the enzyme. And this is mostly work of another student, Roman. So for this, we also use the transformer model that I mentioned before, but we combine that with a variational autoencoder model, which was trained on chemical structures. And we connect the internal representation of the protein from the transformer with the internal representation of the molecule in this VAE model using a small neural network called multilayer perceptron. And then we train this whole system end to end on the database of all sesquiterpene synthases. So here we focus just on sesquiterpene synthases because this is the largest class we have, and we want to make something as a proof of concept. So I'm going to introduce this variational autoencoder a little bit closer. So this is a neural network that takes chemical smiles as an input, and then it encodes it into an internal representation, which is a 196-dimensional vector representing a point in so-called latent space. And then it can decode this representation back into a smiles. And so because you have smiles in the beginning and smiles in the end, you can train this model on a huge number of chemical structures. It was initially trained on 250,000 molecules from the zinc database, but we further fine-tuned it on our own database of molecules that look like sesquiterpenes based on the chemical formula. So that way we get better performance on sesquiterpenes specifically. And so in this latent space, you essentially have a digital representation of the molecule. So we can take a look whether this latent space is meaningful for sesquiterpenes. So we can take some random sesquiterpene, in this case, germacrine A, and we can look in the immediate neighborhood in the latent space, what kind of molecules we see. And just intuitively looking at these structures, we see that the molecules we're getting are quite similar. So it seems like the latent space is meaningful. Okay. So if we make a prediction of some molecule in the latent space, how can we evaluate that it's a good prediction or not? Right? In this case, it's not like predicting the terpene synthase where we know what the answer is. In this case, we might get a molecule that's somewhat similar, but not exactly the molecule that we were looking for. So for this, we use molecular fingerprints. So molecular fingerprints are essentially bit vectors, where you take a chemical structure and you encode some properties of the chemical structure into the bits. For example, here we have a benzene ring, so there's a 1 bit here, a carboxylic group, another bit, and an amino group, another bit here. There are different versions of these chemical fingerprints, different approaches, but it's a fairly standard tool in cheminformatics. And so if we look at the chemical structures using these fingerprints, then if we have the true answer, the true sesquiterpene, and our predicted molecule, we can compare their fingerprints, and we can calculate their similarity using something that's called the Tanimoto coefficient, or also called Jacquard index, which is essentially an intersection over union of the positive bits in the fingerprints. And so this gives us a measure of how similar these molecules are. To give an example, here we have a sesquiterpene delta cation, and here obviously the similarity to itself is 1.0, and here are the similarity values to other sesquiterpenes. So depending on the structure, ranges from 0.27 to 0.7, and here the number is changing based on some modifications that we do to the molecule. So we change a carbon to oxygen, or we remove a double bond, and we add or remove additional bonds. And you can see what are the characteristics? So for example, it seems the fingerprint metric is more sensitive to changing double bond to a single bond than to adding another carbon or another bond. And with this approach, we're getting some predictions, which in some cases are actually perfect. So here's the true answer, and here is the predicted molecule. And these are the same molecules, just different rotation. So these are the examples of the perfect predictions. In some cases, they are not so perfect. So here is the true molecule, it is the predicted molecule. But also, the differences are actually not that dramatic. As you can see, we have one large ring, one small ring. So the molecules actually in some way resemble each other, which is encouraging. We did a complete evaluation of these results, also based on the different architectures of the neural network that's connecting the two main models. And with this multi-layer perceptron, we are getting the best results so far. It's not so easy to intuitively explain what these numbers actually mean, because it's based on like averaging a large number of these Tanimoto scores. But it's actually nice that we are getting a decent number of positive results, and in many cases, actually perfect predictions. We don't really have any other tool that we could compare against. So this method is now becoming our baseline, and we're trying to improve it further by adding more domain knowledge into the whole process. So I would just like to mention here that this project actually was started as a bachelor thesis of Roman, and the thesis was so nice that he got an award for this. So that was a very nice experience. And so this is the end of my presentation. So I would just like to summarize what we did. We developed deep learning models for detecting TPS enzymes and predicting their substrates and products. These models outperformed previous methods based on traditional bioinformatic approaches, such as hidden Markov models. And with experimental validation, we found some previously undetected terpene synthase enzymes with potentially novel products. And so with this, I would like to thank the students who contributed to this project, which is mainly Raman and Roman, who are the machine learning experts, and Teresa and Milana here, who are maintaining our database of terpene synthesis, and Theo, who is working on the synthetic biology applications. And also want to very much thank to our collaborators, Josef Šivec from the Czech Technical University, who has been a fantastic collaborator, providing all the expertise in the machine learning, and more recently, Regina Barcilay from MIT, who is giving up very useful feedback for this project. And I'll be happy to answer any questions.
Great. Thank you, Tomáš, for this wonderful talk. If anybody has any questions, there are already some in the chat. But if you want to unmute yourself and ask questions directly, you can do so. You might want to raise your hands using the reaction panel down on your Zoom screen.
So maybe I can start with
yeah, you can go ahead.
Okay, so the question is, I wonder if you also looked at sequence similarity when conducting chain test split or just similarity of substrates. So we're not looking at substrates, we're looking at the products. So that's how we do the splits. And I think this is a very good point, and we're actually trying to do both now. We're trying to do sequence similarities, kind of phylogeny-based splits, and also the product-based splits. Both have obviously its advantages and disadvantages. And with the product-based splits, we're more looking at like whether the mechanism of the synthase is similar, whereas with the phylogeny-based splits, we're more looking at whether they are evolutionary more related. Ultimately, both are, I think, valid approaches. Second question, do you have an idea of what embeddings work best for these terpene syntheses? I have seen serious variability in language model embedding performance when looking at membrane proteins. I am wondering what discrepancies you might see between different embedding types. Also, what community detection algorithm have you used? Have you considered using a POTS model? Okay, so first the embeddings. We have tried several of these published models. So far, we always came back to the ESM1B model, which gives us in the end the perfect, I would say the highest scores in the predictions. We have also tried dissecting the AlphaFold 2 model and getting the internal representation from the AlphaFold 2 model, and that surprisingly worked worse than the ESM1B. We have recently tried the ENC model, and that actually is the only one that outperforms the ESM1B so far. And regarding the community detection algorithm, I have to say, I don't know off the top of my head. I would have to ask my student what the algorithm he actually used. So another question is whether we have asked Prot GPT-2. I guess that was related to the protein language models. Good question. We have not tried it yet. And next question is, what is the current and expected availability of your tool? So we are writing the first paper. Hopefully should be finished in 2-3 months. I mean, it was supposed to be finished already, but I'm trying to motivate my student. And all of this will be available, including the codes and the database. If anybody is interested in the database that we have, we're happy to open it, and collaborate on it. We're pretty open about the data and the codes that we have. Okay, so that was the last question from the chat.
Yeah, I had one question about these prediction models. So when you do these, when you are able to predict the products of these enzymes, are you able to also go back and infer which residues might be important for generating that activity?
That's a great question. Yeah, so we are also working on this. We're trying to use explainability methods, which are essentially based on messing up the sequence and then looking at how the predictions change. And if you mess up the sequence in a smart way and you do it many times over and over again, you start getting an idea which residues were the most important for your predictions. So we have now a visualization tools that can actually highlight you in the sequence which residues are the most important for giving the result that it gives. Yeah.
There's one more question in the chat.
Okay. Can we use the missing amino acid prediction approach for also predicting for full gene sequence from partial nucleotide sequences with supervised learning model for terpene synthase gene? Okay, I have not tried this. It's essentially, so it's asking about nucleotide sequences, but I assume it's not so important if we're talking about nucleotides or if we're talking about proteins here. I mean, basically, the question is whether we can complete a sequence that is incomplete. Obviously, some information is lost if the sequence is incomplete, and you might probably be able to complete the sequence in different ways and maybe arrive at 2 proteins with different function, right? So this is never going to be a task that has only one solution. But of course, the model could probably give you some prediction, which would be based on the evolutionary context and all the other proteins that it has seen. So yes, some prediction can probably be made. Now, whether you're gonna really get a functional protein this way, that's another question. I mean, there's a lot of work that has been done in recent years on designing protein sequences by like David Baker's lab and others. I would say it's not that simple as just using a trained language model. But if anybody wants to try this, I would be curious what comes out.
Any other questions? If not, then let's thank Tomáš for this excellent talk and for answering all the questions. Please join us next time. And if you are interested in talking more to Tomáš, feel free to wait for a couple more minutes after that.
Sure, I'll be here. And thanks everyone for showing up such a high attendance.
Yeah.
I had a couple unrelated questions, or I mean, they're sort of like more methodological, but sort of a basic level and not relating to the new stuff that you presented. But at one point you